My name is Ahuja, and I'm in third period AP Biology, and I'm also the CEO of Mitochondria Inc. Now, a lot of people ask me what the heck is it that we do here at Mitochondria Inc., and I usually tell them that we're the powerhouse of the cell, but I don't want to lose points off on this test, so let me go a little more in depth. The mitochondrion is a cool cat, membrane-bound organelle, and all of that. It's the site of cellular respiration, making sure you can breathe. Inhalation using O2 to break organic molecules and synthesizes ATP, which is pretty cool. Now let me go back to cellular respiration. There's a real nifty, real cool, easy equation. You take some C6H12O62, add some 6O2 for the heck of the blue, plop on 30 to 32 ATP and some PI. Now you got yourself a reaction. May I? Got 6CO2, 6H2O, some kinetic energy, just to show 32 ATP as a result. And that's the equation. Come join my cool kid cult. Let's take a trip to the kitchen. I sat here one day and began to eat a super carb-rich slice of bread with jam. Ooh, so sweet, so savory, so starchy. The starch just moved its way down to my stomach where it broke into all the glucose monomers. Later, I could literally feel all of the carbon to hydrogen covalent bonds breaking. But first, the glucose was mostly nonpolar, but slightly polar, so that it could travel through my bloodstream and reach every single cell that way. Now let's go ahead and plop myself down on the rowing machine. You know, a muscle cell uses a lot of ATP to function. They allow me to do work like I'm doing right now, doing force and movement. You know, I can literally feel the phosphate groups going through coupled reactions. Now, just so you can keep up, this is number 6. So once I breathe air, the delectable oxygen goes to my lungs. Then, being carried by hemoglobin, it goes to my bloodstream. There's a super high concentration of oxygen in my bloodstream. And then it diffuses into cells. Yo, what's up, cuh? My name finna be a multicellular eukaryotic organism. My homies in Dublin call me Mio. I'm hella efficient. I'm so efficient, finna don't even say the E. Hella efficient. Why? Cause I got those membrane-bound organelles. Like the mitochondria, I do those cool tasks. In the organelles, all my homies can increase bumpage. Finna be increasing concentration inside the membrane. Let's have a little heart-to-heart. The cellular environment. Moment of silence for all the enzymes, electron carriers, substrates, and organelles working so hard in the cell. The nucleus is there, slightly off-center with some nice DNA. Then, the mitochondria. For number 9, the outer membrane is a clean phospholipid bilayer. Next, the intramembrane space contains lots of hydrogen ions. The inner membrane is another phospholipid bilayer but has cristae, which increases surface area for more enzymes on the ETC. On the inner membrane, the pyruvate entry complex and the electron transport chain and the lollipop of life, or ATP synthase, are there. In the matrix is the Krebs cycle. The completely anaerobic process of glycolysis. Takes place in the cytoplasm, needs 4 kinases and a dehydrogenase which are all bouncing around in the cytoplasm. Enzymes bouncing all around, and the probability of bumpage is low? First we need to get in glucose, then we use hexokinase. It don't come close, it takes an ATP and dephosphorylates, and ADP is released and has been created. Now a phosphate is attached to the skeleton. PFK gives an ADP in the gelatin. It's an allosteric regulator for feedback inhibition. Now a second phosphate group is on the carbon addition. You get 2G3P as the product, and now energy output is the next in conduct. Now we start with our 2 carbon chains of 3,1 triphosphate root electrons. You see, it's a very nifty thing called a dehydrogenase, which adds 2 NAD plus become NADH. Now we add in 2 PI. Then we call on a kinase guy who takes the phosphate group through SLP. That's substrate level phosphorylation. You agree? Using the energy with no oxygen, we get 2 ATP twice just to begin. Finally, removing both the phosphates, we get 2 of what we need, pyruvates. Glycolysis, we are now done. What results have you now spun? 2 ATP, 2 NADH, and 2 pyruvates. Moving on to pyruvate oxidation. The products on before move in, no hesitation. Your carboxylase is there right off the bat. Bind to the carboxyl with 2CO2 has that. Dehydrogenase is there too. What do you know? 2 NAD plus becomes NADH for show. Then a coenzyme A, smoosh ACE appears. And acetyl coenzyme A is here for New Year's. Finding the results of pyruvate entry to NADH. As you can see, to CO2 and to acetyl CoA. That's the first 2 steps for the day. Alright, so now we gotta talk about the Krebs cycle. So think of the Krebs cycle like a clock, and there's a bunch of little enzymes floating around. Now, in this situation, I am one enzyme, but think of a bunch of me just floating around. And it's inside the matrix. So everything is super concentrated and all bumping around, so there's a huge probability of bumpage. Now we wanted to bring in one of our most prized and famed doctors. He knows everything about the Krebs cycle. This is Dr. Mitochondria, and this is his lab. Now, we can see here that Dr. Mitochondria is going to show us a lot of stuff, but it's pretty random. But let's just look at some few things. So, the Krebs cycle is like a clock. We can see this because, we have acetyl-CoA going inside, but it's just, let's look at the first 15 minutes, right? So, in the first 15 minutes, there's two main enzymes, dehydrogenase and decarboxylase. Dehydrogenase is going to take away 2 electrons, reducing 2NAD plus to 2NADH. Then we can see carboxylase, which will make we'll take away 2 carboxyl groups to get us 2 CO2. This happens again, but then later we see through substrate level phosphorylation, a kinase produce 2 ATP. And then finally we'll see another dehydrogenase make 2 FADH 2 FAD into 2 FADH2. And so the Krebs cycle is like this random clock with a bunch of substrates just coming out, bouncing all over the place. So, just to recap, we get a few products from the Krebs cycle, which include 6 NADH, 2 FADH2, 2 ATP through substrate level phosphorylation, and 4 carbon dioxides. Now finally we have the electron transport chain. So first we have pencils representing the big guys and pens representing the little guys. First we have flavor protein, then we have ubiquinone, then we have our iron sulfur, or iron sulfate, whatever it is. Then we have cytochrome C, and finally we have the cytochrome A complex. Each of them get more electronegative as you move on. But at the very end, there's the most electronegative guy at all, the air pods, or the molecular oxygen. So each of the 3 pencils channels H plus ions through because they're proton pumps, and they use redox reactions from getting NADH oxidizing NADH to NAD plus, sending the power of those 2 electrons that they're sending through the electron transport chain, which helps pump H plus ions to the intramembrane space, and that's why there's a huge concentration of H plus ions in the intramembrane space. Focusing on O2 for a second, it really wants a lot of those electrons, so it's going to pull everything down towards that end. Okay, this is not oxygen anymore, this is NADH. So it has 2 electrons, but it needs to donate them to the electron transport chain. And the energy of this, which is sending through the electron transport chain, will pump 3 H plus ions through our first flavor protein, because NADH isn't super electronegative, and so it can give them just to the first enzyme. In total, our NADH will give 10 H plus ions through the chain, which will produce 2.5 ATP. If we pretend this is FADH for a second, or FADH2, in total it will give 6 H plus ions, which will yield 1.5 ATP. Now, a gradient is basically when there's a difference in concentrations between 2 areas, which allows them to flow from one another. So in the intermembrane space, there's a high concentration of H plus ions and a low concentration in the matrix. So clearly the intermembrane has the favorability where the H plus ions are more inclined to go to lower concentration, and thus there's a gradient right there. Now this is our friend, the lollipop of life. And so, lollipop of life is kind of like a generator, and where if you get this thing to turn, light will be produced and you'll get energy. The energy in this case is ATP. So this is our intermembrane space, and this is our inner membrane, our matrix. So clearly, when the H plus ions go through this lollipop of life, they help to spin this catalytic knob and produce ATP. Now it takes exactly 4 H plus ions to help spin this and produce 1 ATP. Now the reason it's called oxidative phosphorylation is because at the end of the ETC, that oxygen will collect 2 electrons. And without that, the entire ETC will be slowed down, and so it relies on that oxygen to be reduced to water to channel the H plus ions through. And that's why it's called chemiosmosis and oxidative phosphorylation, because it requires that oxygen. Now, one thing you might have noticed is that in the results of ATP, there's a range. And the reason there's a range is because of these 2 things, that thing, and that thing, which are called lazy Susans. And so there's some NADH just floating around in the cell, but when they come into these lazy Susans, depending on which one they come into, they might be skipping the flavor protein if they come into this one, or not if they go into this one. So depending on it, they might donate their electrons earlier or later. And because if they donate electrons later, they'll give less ATP, or they'll give less electrons, or they'll pump less hydrogen ions, and thus that'll make less ATP. Now there's one final question we're all asking, and that's, what the heck are we actually doing with ATP? Well, in the muscle cell that I talked about earlier, The ATP there is just floating around, and basically an enzyme will remove one of the phosphate groups and attach it to a protein, let's say, in an energonic reaction. That changes the shape of the protein, and eventually what will happen is that protein will get rid of it in an exergonic reaction, and since the phosphate group is now gone in an exergonic reaction, energy is released, and the cell can do work using that energy.
Oh no, we lost our supply of oxygen. Electrons are stuck in the ETC, it's a sin. The electrons get backed up and it all shuts down. Now we can't do work and ATP is gone from town. All the NADH is backed up, number NAD+, but we need to capture electrons, so thus, what we gonna do is called fermentation. It'll oxidize NADH to get a formation. NAD+ is replenished into the cell. Now the process can resume and all goes well. The products in a human cell from fermentation are neat, and NAD plus lactic acid and are all sweet. But if you get too much lactic acid build up, your muscles will get sore and you can't stand up. On the other hand, in fungi and some bacteria, the fermentation process has different criteria. It produces ethanol, NAD plus and CO2. That's why your bread bubbles when you cook before the chew. Eventually, the process can start all over again, since a NAD plus can be used in the next gen. That is all over for this video and the raps. If you enjoyed it, you should give me a good grade and no slaps. But I think the rhymes did slap though. So